Gene sequencing has undergone quite a revolution in the last 5 years or so. If you go back to probably the late 80s, early 90s, gene sequencing was done using a procedure that was called the Sanger method, which was very time consuming because you could only sequence about 1,000 nucleotides at a time. And the process is one where you would take and radioactively label the different pieces of DNA. You have a piece of DNA that's uniform, and you have to copy that DNA, and you'll be able to insert either fluorescent dyes or radioactive material in order to be able to identify what nucleotide, which of the AGTCs was present at each location. And that's a simplification of that. But now the technology has entered a phase called next generation sequencing. And next generation sequencing is means that you do not have to have a uniform piece of DNA. You can take a whole genome and literally take small pieces of the DNA, introduce it into a type of microchip, and then through a process of actually measuring pH changes as the result of addition of bases to individual pieces of DNA. You can determine by individual, let me see if I can simplify this a little bit. You would have a microchip that say has 1,000 little holes in it, and you have 1,000 different pieces of DNA in there. And through some chemical manipulation, you can then start to add pieces of nucleotides to each one of those separate pieces, right? And every time a base pair is matched and added to a piece of DNA, it will give an electrical signal that says this particular base was added to this particular piece of DNA. And of course, it's all computerized, and so the computer is keeping track of all 1,000 of those little wells in there. And those, in well number 1, when we did this reaction, the A was added, but it wasn't added in well number 42. And then when you do a T, it's going to record all the ones where T is added, and so forth and so on, until you get an entire sequence. And then you run computer programs to do matching. You look for overlaps in bases so that you can actually then take all of these short pieces and arrange them into a sequence.